Alrighty, hello everyone, and welcome to Cafe Scientifique from all of us here at SFU's Faculty of Science. My name is Stevie Benish, and I'm the Manager of Outreach and Engagement here in the faculty. If you're one of our regulars, welcome back, and if you're new here, this is your first time, thank you for joining us. We're always excited to have new people. For those of you who don't know, Cafe Scientifique is the Faculty of Science public seminar series. And our presenters, they share kind of what their scientific research is, what its impact is, and it gives you an opportunity to engage with them and ask them questions. So we typically host about 6 of these discussions a year, and they're all virtual and they're also all recorded. So if you enjoy this one and are interested in what else we've done, you can check out on our website and see past ones, or you can share this video or others with family and friends. And we would always love to stay in touch and hear from you, so please feel free to reach out via email anytime. So before we get into it, I would like to start us off on a good note by acknowledging that I'm very privileged today to be joining you from the unceded and ancestral lands of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam peoples. We thank them for having cared for and being stewards of these lands and these waters since long before our time. We look forward to working with them in partnership as we continue to build and grow our communities together. And if you know whose territory you're currently on, I would like to invite you to take a moment right now to reflect on that. And if you don't know whose land you are on, a great resource that we love to share with people is the website native-land.ca. And there you can familiarize yourself with the Indigenous peoples whose land you live and work on. And one more little housekeeping thing before we get into our amazing speaker. Just a couple details. Once again, this is being recorded and it will be shared. There is a live transcript available for your convenience. And following the talk, we will have a Q&A portion. But if you do have any questions throughout, please feel free to make use of that Q&A box if you have any clarifying questions, and we'll try and integrate those as we go along. Alrighty, so now for what we're all here for, tonight's discussion. I am very pleased to be introducing Dr. Nancy Forde, who is a professor in the Department of Physics and the head of a biophysics research group affiliated with the Departments of Physics, Chemistry, and Molecular Biology and Biochemistry, and as well with the Center for Cell Biology, Development, and Disease. So Dr. Ford's research group is fascinated by the molecular level physical properties of our body's building blocks. And tonight, Nancy is going to be talking about the many ways in which collagen supports the body's systems, functions inside and out. And I'm sure, like myself, many of you think of cosmetics and nutritional supplements when we hear the word collagen. And tonight we're going to hear a little bit more about the properties of collagen throughout the lens of physics and its dynamic biological functions, which I'm pretty excited to learn about. So over to you, Nancy.
All right, thank you very much, Stevie, for the introduction. And so let me just share my slides here, and hopefully everyone can see this. I'm really happy to be giving a Cafe Scientifique. It's been a while since my last one. And so thank you to Stevie for the invitation to do so. Thank you to all of you for coming, and I'm happy to engage with you. So please, as you think of questions, put them in the question and answer thing. If they are worth interrupting for Stevie, we'll do so. Otherwise, we can talk about them at the end. But I find it's better to articulate questions as they occur because you don't forget them that way. And Stevie already acknowledged this beautiful land on which we all work and play. And so I'm going to get into my presentation. So my first question, which she also alluded to, is what do you think of when you hear the word collagen? So what springs to mind? If we were in a more interactive environment, I'd have you shout it out, but in this setting, just maybe think about it in your mind, and then you'll see how it compares to what Google tells me. So when I did an image search for the word collagen, here was the #1 hit that came up last night. Something that advertises collagen supplements for radiant skin and strong nails. The next one, another supplement to reduce fine lines and wrinkles. Number three was something that says reduces joint pain. And what you see is bottle after bottle after bottle on these Google image searches of people claiming collagen for some sort of benefit. Eventually, when you get to number 21, or at least as of last night, there was actually something not selling a product, but from a more reputable source, which is the Cleveland Clinic, a medical facility. So I think many people, when they think of collagen, they think of cosmetics, they think of supplements, and so on. But that's not what I think of when I hear the word collagen. Um, Though certainly this is a booming industry. The anti-aging industry was estimated to be over 50 billion US dollars in 2024. So there's definitely a lot of money to be made, but collagen is, I see it through a different lens. And so that's what I'm going to share with you now. So what I think of when someone says collagen. Um, so throughout this talk, I'm going to try to convey to you what my vision of collagen is. And so the outline, just so you kind of have a sense of where we're going, is first of all, I'm going to tell you what is collagen and where is it found. And then I'll talk about how it's made from a biological standpoint, because it is a biological protein. And then after that background, I'll get into some tools of physics that my group is using to learn more about collagen and why these questions are of interest. And then lots of time, hopefully, for questions and answers. Okay, so what is collagen? So collagen is a protein. So proteins are some of the components that make up biological systems. In terms of what it looks like, the structure of collagen is very unique for proteins in that it's a triple helix. And so that's depicted on this slide. But also, if you look at me in the little window where you could see my face, I have my what I call my physicist model of collagen, which is just three rubber tubes that wrap around each other to make a triple helix. So a lot of how I think about collagen is through this particular lens of let's think about it as a triple helix and let's understand some of its properties. Now, collagen is actually a really important protein. If you took all the proteins in our bodies and you put them on a scale, and then you took collagen out and you weighed how much of that there was, you would find that it's roughly 15 to 20% of the total protein in our bodies is collagen in one form or another. And that is a large fraction. Interestingly, collagen is not found in plants. It is only in animals. And it arose in evolution at the same time as multicellular animal life, or so-called metazoans. And so this protein has been, we think it's really important for the ability to scaffold multicellular life. And I'll show you that it forms a lot of what holds our bodies together now. Now, this is my physicist model of the triple helix. This is not to scale, meaning this is not the full size and extent of collagen. If I were to draw this triple helix with the right width to length ratio, it would look something like this line on the bottom of my screen here. So each of these, the most abundant collagens are about 300 nanometers long and 1 nanometer in diameter. And I'll return to those dimensions later. But those are very, very, very tiny. By contrast, the width of a human hair is somewhere between 50 and 100 micrometers in length. So it's almost more than 100 times longer. The width of a hair is more than 100 times bigger than the length of one of these collagen proteins. Okay. So collagen is a protein, and it really holds us together. So it is found in a wide variety of tissues in our bodies, forming sort of the structure that gives shape to these. And in many of these tissues, it actually has quite a mechanical function. So it not only provides the structure of these, you could think of it as a scaffold in some ways, but it also can be a tensile material where its job is to resist strain, as in, for example, tendons shown over here. And turn on my little pointer. So tendons, which are rope-like structures made of collagen, which connect muscles to bone. So every time you walk and your muscle contracts, you're stretching that collagen and it needs to be pretty stiff so that the bone then moves and it doesn't just like super stretchy and you waste all that energy contracting muscles. Okay, so collagen's found in a lot of different tissues. Another picture just to give you a sense of the variety. Um, on the left, you see a number of places. These are listed with different types of collagens, and I'll get into what those are. But all of these have this triple helix structure. And on the right are varieties of cartilage where we can see that collagen is found in all of them. Now, notably, there is no collagen found, or very little, in fingernails or in hair. And so, if you think you're going to take collagen supplements and make your hair beautiful or your fingernails, that's a different protein that makes up those structures. That is called keratin. And I'll talk a little bit more about that later, but it is not collagen.
I think this is actually a perfect time for a question that just came up
Yeah.
about where can vegetarians get their collagen from?
Aha. So if you're a vegetarian, you will not be ingesting collagen. And by the way, gelatin is a product that comes from collagen. So you can't eat marshmallows or gummy bears unless they're vegan if you're a vegetarian. But you don't need collagen in your diet. So cells will make the collagen. I have a slide on that in a little bit how it's made. So as long as you eat a well-balanced diet and get lots of protein and everything else, your body will be able to make collagen for you. Yeah, great question. Thanks for interrupting with it.
Yeah, of course.
Okay. So it is perhaps not surprising if you think about all these different parts of your body where collagens are found, and really where they help to template the structure or form the structure and so on, that if you get a mutation in one of these collagens or you change the identity of part of this protein, that problems might arise. And indeed, there are various genetic collagen diseases that are associated with different parts of the body, different types of collagen. So in the left side, all these call something or others are different types of collagens that are found. And all of these have been identified as causing diseases. And on the right are some of the more abundant collagen diseases. Um, so a lot of these have to do with defects in the structure of tissues, in the mechanics of the tissues, and so on. So understanding the mechanics of these collagen-based structures is really important to try to understand how you function and also how dysfunction arises. And so if we look at these collagen diseases, yes, there's a lot of different proteins. If we zoom in on osteogenesis imperfecta or brittle bone disease, and we look at this triple helix of the collagen that is associated with osteogenesis imperfecta, collagen 1, which is the most abundant one in our bodies, forms the bases of our bones and tendons. What this shows, these little sticks represent every location along this triple helix where researchers have identified a change in the chemistry of the amino acid, the constituent at that location that can lead to OI. And what you can see is there's a lot of different places where if you change one of the proper amino acids to something else, you can get this disease. And depending on which one and where, it can be quite severe. Okay, so let's go into how collagen is made up so that this is perhaps a little more understandable if you're not used to thinking about proteins. So how does collagen made in our bodies? Well, it's made, at least initially, very similar to any other protein in our bodies. So step one down here on the bottom is the DNA starts this all off. And in the DNA, you have genes, and a gene is like a stretch of letters in the DNA alphabet, A, T, G, and C. And it will say, oh, these are the instructions or the recipe for making a particular protein, for example. But in order to make a protein, the first thing you have to do is copy that. And so that's step two, is making an RNA copy, which is another long string of letters stuck together, 4 letters. And then from there, this mRNA or messenger RNA gets translated into different components called amino acids that get stuck together. And so if you read along this mRNA instructions, you look at a group of 3 of these, for example, a G, and a U, and that tells you, oh, there is a glycine there. So we have amino acids are the building blocks of proteins. And these first 3 steps happen for any protein in our bodies or in bacteria or in plants or anything. What's a little bit different about collagen is that after this chain has been put together, there's some additional chemical modifications that happen. And so there are certain enzymes or little machines that go in and do some chemical modifications to some of these to make them a little different than the original building blocks of the collagens, okay? And then while those modifications, those chemical changes are happening, the 3 chains somehow find each other and they zipper up to form the triple helix. So this step 4 being written twice is not a typo by me, but it's really because we think these 2 are sort of happening at the same time. Okay, and so that's a little bit different than a lot of other proteins, which are made as a series of amino acids, but they'll probably fold into a more compact 3-dimensional blobby structure rather than this very long thin chain that's actually 3 chains that are all finding each other and wrapping up. So all of this happens inside the cell, inside of our cells. And then in order for collagen to have function, it needs to get outside the cell because where it's found is outside of our cells, forming these scaffolds of the extracellular matrix or of connective tissues like the tendon and so on. And so outside of the cell, once these triple helical proteins are secreted or spewed out of the cells through their proper channels, collagens assemble into higher order structures. And really, these are the materials that we see when we look at connective tissues, are these assemblies. So think of the collagen triple helices, these individual proteins, as the building blocks for making higher order structures. You could think of them as Legos, but they're not really like Lego bricks. It's more like threads, rope. So if I have my rope here, I have a rope and it's made up of tiny little threads. And those little threads all come together to make this very, very strong rope. So that's the sort of picture you could have about collagen assembling into higher order structures, which happens outside the cells. Okay, so fibrils or fibers, these rope-like structures are the most common structures that collagens make. And so many of the collagens, like the ones that form our tendons and bone, form these fibril-like structures where the collagens really sit together side by side like threads. What's different than a rope is it's highly ordered so that you see a pattern. When you look at a fibril with electron microscopy, the axis goes left to right, and you can see this beautiful structure. That's because of the high degree of order of these collagen threads. And other collagens like type 4 collagen, which tends to be found in the kidney and other organ linings, it tends to form more of a network-like structure. So even though these have similar triple helical structures, slight differences in them can cause them to form different higher order structures. And these can form not only in a biological setting like our bodies, but we can make these in the lab. And so the pictures at the bottom here that were taken with electron microscopy are showing you the types of structures that can be formed in the lab when you take these isolated triple helical collagens and you mix them together the right way. So the information for how to assemble is encoded in their composition. Okay. And then these types of structures then go on to form higher order structures like tendons and so on. So that's kind of some background on collagen. And I have so much more I could say about this fascinating protein, but I thought I'd spend some time telling you a little bit about what my group is doing as well to learn more about it. And so really what my group's interests are is trying to understand what is the interplay between collagen's composition and chemistry, its chemical components and environment, and the mechanics of the protein and its stability. And so when we say chemical composition and we think about this triple helix, we could mean, oh, is this type 1 collagen, type 2, type 3, type 4, which have slightly different arrangements of these amino acids in their sequence? Or it could be a chemical composition in that how does aging affect this? So as we age, our collagens get chemically modified and changed. And they accumulate these chemical changes gradually with age. We can mimic that in the laboratory. And so we've been looking at how does that affect mechanics? I'll tell you a little bit about that. And then also, as I showed, there are mutations or changes at particular locations of the triple helix that can give rise to these connective tissue diseases. And that's a direction in which our research is heading, is getting these collagens from collaborators to look at. We're just starting to do that, so I'm not going to tell you anything about that yet. Why do we care about mechanics of collagen specifically? Well, not just because we're physicists and mechanics and forces are really relevant, but also they're relevant to function. And so I love to use this example from Terry Fox, who was a former SFU student before his Marathon of Hope. And think about the applications of collagen mechanics. For example, we could make better prosthetics and re-engineer tissues to replace damaged tissues in the bodies. If we think about this tendon, for example, that is forming this connection between the muscle and the bone. We look at it, you can see here's a nice drawing showing sort of the hierarchical assembly from triple helices to these fibrils to higher and higher order structure. If we understand the function of these building blocks, that can help give us some insight into how these tissues function and how they're affected by aging or disease. And also, what is quite interesting is there's a huge field developing called mechanobiology that is looking at the interplay between forces and mechanics and what happens to cells. And what is being learned by this community of researchers is that depending on their mechanical environment in which a cell finds itself, it will respond very differently. And so stem cells, for example, will turn into bone cells preferentially when they're on a stiff surface, or into fat cells when they're on a squishy surface. The forces involved with development of a fetus can regulate what happens. And also in diseases like cancer, the mechanics of this environment that surrounds cells can in turn influence how they behave. So we're really trying to get down to the building blocks and understand how they function and their structure, stability, and mechanics in order to take a bottom-up approach to understanding then how that might feed into these properties of higher order structures and tissues. Okay, so one of the first questions that we could think about is if we're thinking about this triple helix, that our collagen structure, how easy is it to bend and flex? So is it a rigid rod or is it a more flexible structure? This could influence how easily it can align and pack with neighbors to make these rope-like fibrils. So to give a different picture for that, the question is, are collagens more like cooked spaghetti or raw spaghetti? Are they flexible or are they rigid? And so that is a really good starting point for understanding mechanics of these things, is just what is their inherent flexibility like? And then we can see how it's modified. So the challenge is collagens are not as big as pieces of spaghetti that we can look at with our eyes and just characterize whether they're changing direction a lot or straight lines. And so to give you an idea of the scale of collagens, I'd like to show a video that's prepared by CERN, which is a particle physics lab based in Switzerland. And they're prepared a video to take us on a journey from like a human, and then they zoom in on human hair and they go to smaller and smaller scales. So I want to remind you first that collagen is not found in hair. So we're not going to be looking at collagen, but I have not found a video like this about collagen structure. And I think that the keratin proteins that make up collagen are very similar in many ways to the assembly of collagen. So let's just take a look at this video, and we're going to zoom in on human hair, okay? To give ourselves a scale of proteins. How big are proteins? And so as this video is zooming in further and further, you can see the scale bar along the bottom. So we're down to 100 micrometers in diameter. So that's the diameter of a human hair. We're zooming in further, we can see what were cells. And so keratin has come from inside cells. It doesn't get sent outside. That's how it's a bit different. But you can see even then that there's layers of structure, those macrofibrils. We're zooming in, we're seeing that there's a smaller scale structure, microfibrils made up of these proteins, protofibrils even smaller, 2.8 nanometers diameter, and then we get to the scale of individual keratin proteins, which are not dissimilar in diameter to collagen proteins, okay? When we zoom in further, we see the proteins are made up of different types of atoms that are bonded together. And then this video is great because it keeps going past the atoms. Here's a carbon atom and the electrons that are orbiting around the nucleus. This is why this came from a particle physics lab, as they really want to get into the particles that make this up. And now we're trying to zoom into the nucleus. And you can see from the bottom, there's just so much space taken up by a carbon atom, and most of that's the electron clouds. We finally make it to the nucleus, which has protons and neutrons. And then inside of those, they're made up of quarks, okay? So lots and lots of orders of magnitude of size in this video. And what I love about physics is physics studies all of these scales and beyond, right? Beyond us to the galaxies and the cosmological horizon. What I'm interested in are proteins. And so just to remind you from that video of the keratin, these were structures that were about 2 nanometers in diameter. They're also long and slender like collagens. Collagens are even narrower. The triple helix is about a nanometer in diameter, and then 300 or 400 nanometers in length, depending on which type. And what's really important to note is that these size scales are too small to see with a light microscope. So if you put collagen proteins or keratin proteins on a microscope slide and you looked at it with even the best light magnifier, you would not be able to see it. It's too small. We're up against something called the diffraction limit of light. And so if we want to get to this question of, is my collagen like raw spaghetti or cooked spaghetti? And we want to understand what are the conformations it can adopt, and does it look like straight lines or more flexible things? We need a different tool than light microscopy. So how can we visualize this? Well, a different approach to sensing this is sensing by touch. And so this is the basis for an instrument called an atomic force microscope, or AFM. And this is a photo of an older AFM that we used for our initial studies, and a schematic of this. And what I'm going to do is show you an animation that illustrates how this very sharp probe, sort of like a record needle, for those of you of an age to know what that is, can sense and feel what's on this surface below it. So what we want to do is we want to put collagen molecules on a flat surface and then kind of use braille like feeling for where they are sitting on the surface. So the way this works is it scans one position at a time and senses, oh, where's the surface? Where is the surface? Where is the surface? And it's very good at measuring undulation. So if there's things that are sitting up above the surface, we can measure their height, because we feel resistance at a higher position than when something is flat, okay? So this is the basis for an atomic force microscope, AFM. Really, it can sense forces on the atomic level, but our approach is just simply to use it in the way that you can think of with braille, that you can feel things sitting on a flat surface. Okay, So, what does collagen look like in such an instrument? Well, here's a couple of images. So what we do is we deposit a solution of collagen on this very flat surface. We use mica. It's a mineral that can peel off and be atomically flat. So it's really, really flat. And these collagens that are a nanometer thick at most when they're surrounded by water are taller than that surface. And so in the image on the left, what you can see is a bunch of yellow squiggles, and each of those is a collagen protein, one single collagen protein. This turns out to be collagen 4. It's a little bit longer than the other ones, but it's a really beautiful image recorded by my former PhD student, Al Shire. And if you zoom in, you can even see like one individual collagen and its path. It is not a raw spaghetti, right? It is flexible. Okay. So one of the first questions that we wanted to ask was really, how flexible are these collagens? Clearly not raw spaghetti, more like cooked. But does the flexibility depend on what type of collagen it is? And so this project was started by PhD student Naghmeh Rezai and continued by an undergraduate student, Erin Lyons. And they looked at a number of different types of collagens from different sources. And types 1, 2, and 3 all form these rope-like fibrils. These are example images of them. And you can see none of them, like on average, they don't look like rigid rods, but they also don't look like completely collapsed structures. Okay, so physics comes in with the instrumentation, but physics also comes in with the analysis. So how can we quantify this? We want to put numbers to this, and we want to be able to determine, are these similar or are they different? And so to do that, what we have to do is trace the backbones of these chains in our images, and then use some models from physics to interpret those backbone trajectories. So Naghmeh developed a great chain tracing algorithm, and undergraduate Matthew Schneider used machine learning to automate the process and basically identify the coordinates of the chains in these images. From there, we start to get into more theoretical physics models and say, okay, what are we going to do to interpret these coordinates? And we use a model called the worm-like chain, not super important, but we're basically, it gives us equations that we can use to understand the flexibility. Here they are. You didn't know you were going to get equations in this talk, but there we go. I have to put in equations because this is physics. Um, but what I want to show you is just that they actually work to describe the data. And so that's what I'm intending to show you here is, look, circles are the experimental data, lines are this theoretical model, and they agree with each other really nicely. And they allow us to get a parameter, which is called persistence length. And that tells us whether this thing is really floppy or really rigid. Long persistence length means more rigid. Okay. And Stevie, yes, there's a question.
Yes, yeah, there's a question just from the last slide about the white ends, or I guess the slide before, the white ends on the collagen molecules. Is that
Mhm.
just an artifact or is that something specific?
On this image, perhaps.
Yes.
Yes. So this is special. It's only on the type 4 of the images I've showed. And yeah, this is a part of collagen that's unique to this network forming collagen 4. It has a blob on one end, so a globular domain. So when I said that a lot of proteins from this chain of amino acids fold up into sort of compact three-dimensional structures, part of this does that. And then you have this long, thin thread coming out from it. So it's very astute for someone to notice that. That definitely it's unique to this one and how it's processed. It's very handy for us because it gives us an end marker and we can distinguish one end from the other. Whereas for these other collagens, they've had their globular ends cut before they get secreted from the cells and form these higher order structures. So we don't know which end is which. Um, Not necessary for this analysis, but it's really important for some other things we've done.
Yeah, that's super interesting. Thank you so much.
Yeah. Okay, so the key for this is we have a nice physical model and the numbers, look, we have two different things we can read out and they agree with each other. And when Nakme and Aaron looked at these different samples that I mentioned, different types of collagens from different sources, what they found is they all have approximately the same persistence length or flexibility. It's all around 90 nanometers or so. Okay, so persistence length is just how long along the backbone do you walk and you keep going the same direction? Let's say it's 100 nanometers. That's a third of the way along one of these collagens. So they're not really flexible, not changing direction super fast, but they're definitely not like raw spaghetti. So this tells us there's not an enormous impact of individual amino acids on these flexibility and mechanics. However, I did show you those images of type 4 collagen, the ones with the blob on the end. They form different structures. They don't form these fibrils, they form networks. And so when Alla looked at the type 4 collagen and analyzed it, what she found was it's considerably more flexible. It bends much more easily than these collagens that form fibrils. And so what we can sort of take away in a big picture context from that is that the collagens that form fibrils start as more rigid, and the collagens that form networks start as more flexible. And we think that those properties might have something to do with how easy it is and how energetically favorable it is to form these different types of higher order structures. Okay, so that's one lesson that we've learned about composition, and we've dug into this in quite a lot of gory detail, but I'm not going to go into that for the purposes of this talk. Instead, I'm going to give you a different example of chemical changes and how they influence collagen mechanics. And this was a project that was initiated by another undergraduate in the lab, Daniel Sloceris. And Daniel's super interested in aging. And so he implemented a chemical treatment of collagen that is commonly used in the scientific field, where if you incubate collagen with sugar, you will modify it chemically. And there's different types of things that can happen. Things can get stuck on the sides of chains, or chains can get kind of stapled together, these so-called crosslinks. We can do this in the lab. So he could incubate just mix collagen proteins and sit them in a ribose solution and have this happen. And what many others have shown is that this is actually a reasonable model for the types of modifications that happen in our bodies as we age. Okay, so collagen is pretty long-lived in many of our tissues. In our cartilage, for example, the half-life has been estimated at over 100 years, meaning it doesn't turn over very quickly unless it needs to be because of damage or something. So collagens sit around for a long time. They have a lot of opportunity to pick up sugars from the bloodstream and have these very slow chemical changes happen. This can also happen in diabetes. If you have uncontrolled blood sugar and you have too much around, you get these types of modifications. And so cleverly, these modifications have been named by the community as advanced glycation for sugar, end products, or AGEs. So that's a clever little acronym from science for you. Okay, so what happens to the mechanics of the single collagen proteins if they get aged? Are there any effects? So Daniel performed the same type of analysis with atomic force microscopy and compared aged or glycated, sugar-treated, and non-aged or unglycated collagens. Again, looking at these images, you can't tell a difference between them. But by scanning many, many collagens and taking these models and applying them, what he could see is for every pair of experiments he did, the aged collagen looked stiffer than the one that had not been aged, but just sat for the same length of time without sugar. Okay. And so this really told us that, hey, these chemical modifications that we accumulate during aging are stiffening the proteins on the individual protein level. People hadn't known this before. There had been studies of higher order structures that were stiffening, but there's a lot of interactions between the collagens in like a rope-like structure that could be responsible for stiffening. What we found was that it really is important at the molecular level, the single protein level too. Daniel also looked at other influences of aging on these collagens and the properties of them. For example, their ability to form these higher order structures, these fibrils. What he found was if he incubated them without sugars, they could form fibrils. Here's a light microscope image. So we can't see the individual proteins, but when they assemble into these thicker ropes, we can see the ropes with light. But if we took the collagens that had been sitting the same length of time, but in ribose, they did not assemble anymore. So these modifications seem to be preventing them from forming higher order structures. He also found that if he looked at the collagens that had been treated with the sugars versus not, they melted at a lower temperature. So here these yellow lines are showing how much triple helix is there measured with this spectroscopic technique versus temperature. And you can see that you're going from triple helix to unfolded at a lower temperature for the chemically treated ones than for the untreated ones. So we're destabilizing the triple helix. And finally, he did an experiment with a biochemical technique where he took the collagen and he tried to cut it up with an enzyme. So basically, think of it as scissors. And these scissors, called trypsin, can only cut single chains. They can't cut the triple helix. So if you have a nice stable triple helix, can't cut that. But if your collagen is destabilized and starting to unfold a little bit, trypsin, these scissors can get in and cut it up. And what he found was that he saw a lot more cutting, here shown as loss of these bands in a gel, when he had treated this with the ribose compared to not. So all of these indicate that collagen is getting chemically changed, its stability is changing with age, and its mechanics is changing with age. And so we're interested in sort of taking that and learning more about how that affects these higher order structures, as well as looking more into the individual changes. What I'd like to do next is tell you a different aspect of mechanics of collagen, and this is to do with stretching it. Okay, so thinking about our tendon, when the muscle contracts, it pulls on that tendon and stretches it. Many of our tissues, or connective tissues, are under load. So what happens when we take individual collagens and stretch them? Not the whole tendon now. Let's just look at a single protein, grab it and stretch it. So if we think about this triple helix, we stretch it, we could imagine what might happen is it tightens, it becomes harder for the chains to come apart. Maybe nothing happens, or maybe we destabilize it. Maybe it unwinds a little bit or something when we pull it. There's evidence from other people in the literature for all of these options. And so a PhD student in the lab, Mike Kirkness, came and said, you know what, I'm going to use these scissors, the ones I just told you about that Daniel had used. Mike used them first in this particular experiment. And he thought, okay, if I add these scissors to the collagen, what should happen? Under no force, there should be no cutting because the trypsin can't access a single chain. This is a stable helix, can't cut. We need to have it open. So if it doesn't change structure under force, nothing happens. If it tightens, we still can't cut it. But if force, when we stretch it, it loosens it, then we should be able to start seeing the cutting. So how do you probe that? These are teeny molecules that are nanometer in diameter, 300 nanometers long. We can't see them easily with light. And how do you grab them and stretch them? So one way is to attach them between a surface and a microsphere, so a particle you can see under the microscope. Okay, so there's chemistry you can do the ends in order to grab onto this. And then imagine this bead is heavy, it can feel a force and be pulled down by that gravitational force. What's nice about this is you can parallelize it, and so you can attach a whole bunch of beads to your cover slip using collagens and look at them in a microscope. And so each one of these then could be a bead pulling by gravity on a single collagen protein. Then how do you do the experiment? Well, if the trypsin comes and cuts this, the bead is going to fall because of gravitational force and it will leave your field of view. So over time, what you would expect to see is if your collagen is getting cut, you gradually lose beads because they fall down and they're no longer in focus. Okay. The problem is if you want to see how it responds to force and you want a different force, then how are you going to do this? You can use bigger beads or denser beads. That's really tricky to do all the chemistry all over again. But another way to do this is to take your sample and spin it, right? So picture one of those rides at the PNE, right? Where you go around and around and around, and the faster you go, the more force you feel like, centripetal acceleration. You're getting pulled out. So with this one sample, depending on how fast you spin it, you can exert different forces to pull on the collagen and stretch it between its ends, the one end that's tethered to the surface and the other one that's on this bead that's getting pulled by this acceleration. Of course, to do this, you have to spin your sample and you want to look at what's going on. So you actually have to spin your whole microscope. And that is the basis of a centrifuge force microscope. So Mike built this in our lab. This is our battered up version after he'd done a lot of experiments with it. And it goes in its little protective housing into the bucket of a centrifuge. The centrifuge is a commercial instrument that spins these buckets around, just like the rides at the PNE. Usually it's used for biological things. But in this case, it's spinning our microscope. And Mike built this to have a wireless connection. So it doesn't have any wires. There's a LED light source at the bottom that's battery operated. There's a camera at the top that's battery operated. And it sends out a signal that's received by this receiver that sits outside and sends a signal to a laptop. And so as you're sitting there at the laptop beside this thing that's spinning around, you're able to view what the microscope is seeing through the objective lens. Okay. Now,
We just have a quick question about
yeah.
this experiment.
Mm-hmm.
So I thought this is a good time to interject. Just regarding, says gravity can be very weak. Wouldn't an electric charge be a better experiment?
Mm-hmm. So in this experiment here, I'm going to just go back so I'm not making people dizzy. What we're relying on is that the beads we're using are heavier or more dense rather than the water. So they are going to feel a sinking force. So we're mostly exerting, the force is being exerted on this bead that's at the end of the collagen, but because it's attached to the collagen, it's stretching it. So that's the thing we're exerting the force on. The gravitational force is small, but it's not as small as what the electric force would be on that particle. We have also done experiments in our lab where we use optical forces to grab onto beads that are attached to the end of collagen. That's a technique called optical tweezers or laser tweezers. You can also use magnetic fields and apply magnetic forces to these beads. That's a technique called magnetic tweezers. These are all techniques, well, magnetic tweezers and optical tweezers are techniques that are quite widely used in single molecule biophysics, which is actually a really big community around the world. This centrifuge force microscope is a bit of a newcomer to the field. And the technique was developed, as I have written down here, I should mention, by Ken Halverson and Wesley Wong. They first built one with like a meter long kind of spinning rebar almost. And then when Mike came, he said, I want to miniaturize this and put it inside a centrifuge. Ken and Wesley have subsequently also done the same thing. And so we have a couple of different versions of these CFMs floating around.
Awesome. Thank you, Nancy.
Yeah. So, I mean, you need a centrifuge for this, but most labs have one. We borrowed one from molecular biology and biochemistry to start, which was awesome. And but the microscope itself is incredibly affordable. So he built this for $500 in parts. We can see lots of molecules at the same time, have a really wide range of forces we could exert by spinning at different speeds. So this is a really high throughput method for these types of experiments. So let's see what happens. Okay, so I'm going to show you. This is a image from the microscope. It's recorded while it is spinning. In this particular case, it was at 800 revolutions per minute, so going really, really, really fast. That works out to a force of about 9 piconewtons on each of these beads, which sounds really small. Pico is like 10 to the minus 12, a trillionth of a newton. But on the biological molecular scale, this is the relevant range of forces. And so let me remind you of what we want to look for is if these trypsin, these little Pac-Men shown in this picture, are able to cut the collagens, then what we should see is that the beads disappear over time. And if the collagen is not cut by trypsin, then what we should see is the same number of beads versus time. Okay, so I'm going to play the movie for you. I think it's going at about 10 times real time. There's been some processing. So you're just going to see sort of snapshots evolving. So watch the beads and remember this is in a microscope that's spinning at 800 RPM. Okay. And what do we see? We see beads disappearing with time. Okay. So that's suggesting that what's happening is that the collagen can get cut by this trypsin when we stretch it. So maybe it's destabilizing. Okay, but we have to be really good and do lots of controls because we're scientists, right? We have to make sure we're doing well-designed experiments. So what we can do is we can compare what happens under this force over 20 minutes. So there's 2 snapshots at the beginning and after 20 minutes with the spinning versus what happens if we just left it sitting on the bench and looked at it with a regular microscope. So the force is very small. You can see hardly any beads are left, are gone, sorry, in the top. So it really seems like force is important in this experiment, that this disappearance of beads doesn't happen unless you start pulling on them. We can also take snapshots at the different times and count the number of beads, go what fraction of beads are remaining, and without any force, we see very few beads getting lost over time, whereas when we have high force, we see beads disappearing rapidly. So that again suggests, woo, something's happening with force. But we need to make sure that what's actually happening is that's because of trypsin, because the connection, this collagen is getting cut, and not just that we're yanking this off the surface by pulling on it. So we have to do another set of experiments where we repeat all of this, but we don't have trypsin, we don't have these scissors, and we can just look at how much is detaching from the surface because of the force. And that's the open circles. So you can see it goes slower. There is still some bead loss when we have no trypsin. So some of these detachments are happening because the things are getting yanked off the surface. But having these scissors, this trypsin there to cut them makes it go much faster. And so once we've corrected for that, what we see are these rates that we get. And the key take home from all of this is that we've enhanced the rate of collagen cleavage about 20 times because of the application of force. So force is pulling on the collagen has done something that makes it get cut more easily. So if we look back at the picture, what that tells us is that when we apply force, we're loosening the triple helix, unwinding it perhaps somehow to make it easier to cut. Okay. And these are not based on a single run. We've done lots of experiments, but we're getting thousands of molecules, individual measurements to contribute to these data. Okay. So just as sort of a point of curiosity, we have our centrifuge force microscope, we have a space shuttle, we have kudos to the Olympics that just happened to luge and NASCAR. And if we were all in person together and I could get you to raise your hands, I'd say, which of these is experiencing the highest acceleration? So give that a thought. And I'll tell you that the highest G-force humans can sort of withstand in a continuous way is on the order of 5 Gs. Space shuttle was about 3. NASCAR is about 3, though F1000 apparently can get up to 5. That's at the turns because you're getting a large centripetal acceleration. Luge is higher, it's 5 Gs at these turns. But But for us, our highest G-force can be 1000 G more for our microscope. So we've built something that's really quite remarkably stable under these, or Mike built something highly stable under these accelerations. Okay, so in the last couple of minutes, I'm just going to share with you one more vignette about collagen that has fascinated me since I first learned about it. And then we can open up for more general Q&A. And that is, if you take collagen, these proteins, these triple helices, and you heat them up, they will fall apart. The three chains come apart. They're not held together by chemical bonds like covalent bonds. So you can heat it and they'll fall apart. But then what happens is when you cool it back down, it doesn't do like my model does and form a nice triple helix. Instead, it forms gelatin. So no one has actually been able to take a pot of collagen, heat it up, cool it back down, and get back collagen proteins. Instead, you get a gel. And that's the basis for things like bone broth. You take your bones, you boil them, the collagens come off from the bones, and then you cool the stock and you get this nice gel thing, right? That's because of the collagen turning into gelatin. Now, what is most remarkable is trying to figure out the temperature at which this happens. It's so-called melting temperature. How hot do you have to get collagen in order for these chains to fall apart? Clearly it's below boiling temperature, 100 degrees, because we can make soup broth that way. But it turns out if you have these proteins isolated in solution, it takes less than body temperature for them to fall apart. So let's just think about that for a moment. Our body temperature is about 37.5 degrees. If you take collagen and you sit it at 37 degrees, it will fall apart. But I told you, collagen is the most stable, like it's the building block of our tissues, right? It's what holds us together. It's not gelatin, it's collagen. All of this talk has been about these triple helices. So how can this be? Why are we not puddles on the ground? And what is it about this structure that is so close to this threshold of stability? So my hypothesis is that it's really close to the stability threshold because then it's very sensitive to its environment, and maybe changing its shape and slightly unwinding can allow it to convey information to cells. And the cells use that as a way to sense what's happening outside. That's something we're working on. Okay. So Ella, who worked on this beautiful project with type 4 collagen and learned how to harvest it from mouse cells and so on, she decided to investigate this. And so here's, again, a reminder of what collagen 4 looks like when it's been sitting at room temperature. So nice and stable, the blob on one end, and then this little chain below it. So what Ella did was to place it in a test tube at 37 degrees Celsius, and then take it out at certain time points, put it on this flat mica and image it with the AFM to see what does the collagen from solution look like after it's been sitting at 37 degrees. Okay. So after 30 minutes, she gets images like this, where you can see we've got these blobs on the end. And a lot of these look just like regular collagen 4, but there's some that look very collapsed. After 90 minutes, you see a lot more of these collapsed structures relative to the amount that look like proper collagens. After 200 minutes, there are very few full-length chains remaining. Most of them have collapsed. And so really, this is seeing is believing, right? That's the header on this, that we see this. It's not just something that's a measurement made in a lab, but you can visualize the fact that collagen is not stable at 37 degrees. These things are falling apart with time. Now, what Ella also did was to take these collagens and cool them back down. So if we did that in the big solution, we expected to see gelatin. I said, we can't make these triple helices again, but it turns out you can. Okay, so here's the heated sample zoomed in on one collagen from one image. If we look at another image of a different collagen after it's been sitting at room temperature for an hour, looks like the three chains are starting to zipper up The length of this backbone is getting longer. And if she let it go overnight, these things are starting to look a lot more like what we started with. So why is that? Why can we do this? It turns out that in the collagen 4, there are these special chemical bonds between the chains at one end, the end away from the blob, the other end, that are formed through sulfur residues, cysteine residues. There's sulfurs, and they can sort of make chemical staples. We like to call them disulfides. And so you have these staples that are acting kind of like the clip at the end of my collagen model here and holding the three chains together at one point. So even if they fray apart elsewhere, these are held together. And we think that's the nucleation point that allows them to start rezipping again and gradually increasing the length of the helix from one end to the other. Even after overnight, we did not get everything folding and we didn't get it all folding correctly. So it's very, very slow, but it is possible. So Alla wanted to test if these staples were important. And so you can treat the collagen chemically and remove them. So you basically remove your clamp from one end. And when she did that, it looked fine at room temperature. But when it heated, the things fell apart. And after cooling overnight, they did not come back together. So they really need these staples at one end in order to allow the refolding to happen. She looked at other types of collagen, so collagen 3, which has these staples. She could refold that overnight, but collagen 1, which lacks them, did not. And so in her recent paper, she looked at a lot of characterization of these unfolding and refolding pathways. And we think we've got a better idea of some of the chemical determinants of what allows these things to happen. Okay, so I'm going to wrap up now. I'm hoping that after this talk, when someone says collagen to you, maybe you don't think first about nutritional supplements or beautiful skin, but you think of how cool and fascinating this triple helical protein is, the structures it forms, I will say nutrition is important. Your body needs to be able to make collagen. And one of the most essential things you can eat to help it make healthy collagen is to take enough vitamin C. And I'm happy to tell you more about the vitamin C story afterwards, but please do eat proper foods and a well-balanced diet. I'm not a nutritionist, so that's about all I'll say on that. And finally, just before I answer whatever questions you have, I really need to give full acknowledgments to the people who did this work and the money that paid for it. So as I went through, I highlighted the students who've led these different projects in the lab. This is my current research group in this photo. And we've had some collaborators for collagens to send us samples of some of the proteins I talked about. And most particularly, I'd like to acknowledge funding from NSERC and from taxpayers like you who fund NSERC, which is our national funding body. We don't get nearly enough money for science funding from NSERC. We really need a lot more to help us do good research, but we are very grateful to the funding that we do get from them that allows this to happen. And I would also say that the scientific community is incredibly generous. So, for example, the AFM that I showed you the photo of at the beginning broke down partway through Alla's PhD thesis just after she got back into the lab for COVID. The instrument died. And Emily Cranston at UBC was incredibly generous, letting her access their AFM to complete her PhD research. We're very happy we got funding from NSERC to buy a new AFM, so we're all good to go now. But with that, I'll wrap up and happy to answer any questions.
Alrighty, we actually have a lot of questions, which is great. Um, so let's get to it. Okay. I think I've got them all on a list, but if anyone else has more questions, please feel free to put them in the Q&A or chat. And then if we end up having too many questions that we don't get to them all by time 6:30, I will share them with Nancy and then we will send out those answers via email afterwards. So, okay. So first question
Uh-huh.
is, does the body differentiate between dietary collagen and the body's own manufactured collagen?
Yeah, so I am not a biologist. I have no training in biology. I've learned as I've gone. But I don't think that the dietary collagen does very much for us that any other protein wouldn't do. So we ingest protein and it gets broken down in our stomach by enzymes, these little scissors like the trypsin I mentioned. There's ones called pepsin and other things that break down the food into the little individual bits that then get given to cells to help make the proteins from these individual components. So when you eat collagen, so first of all, the supplements are not these 300 nanometer things. They're more like chunks of little bits of gelatin fragments. Um, and I think they're just treated like other proteins would be. And so there was a really nice article actually by that a reporter from the BBC recently all about nutritional supplements and how most of the studies don't do super well controlled things controlling collagen to other proteins, but use other things for comparison.
Great. Okay, next question. Can collagen be delivered to the body transdermally?
So I think so, but it depends what what one wants to do. So the collagen proteins that I talked about, you could put them in fluid and sort of inject them in principle, but they'd need to be able to assemble into higher order structures to be useful. Otherwise, they'll fall apart. As we saw at 37 degrees on their own, they're not stable. They're only stabilized by being in these higher order structures. Um, and so, yeah, there are people who are working on peptides, shorter pieces of collagen-like things to see if those can be used in a um, transdermal sense. I'm not an expert on these things, so I don't want to comment more than that.
Alrighty. So there's a couple kind of similar questions. First being, are there any collagen-like proteins in simpler multicellular or unicellular organisms? Do they exist elsewhere?
Yes. So when we look through evolution, well, we being not me, though Alla has done this, she did that with these staples that I talked about to find out where they were in evolution. And when others look, what you're looking at is the DNA usually, and you're looking for the segment of DNA that encodes a protein. And you can see that there's collagen-like sequences that go back in like organisms that have been around for a very long time. And these seem to go back to the dawn of multicellular animal life. And collagen 4, this network forming collagen, seems to maybe have come before the fibril forming collagens. So they're definitely in animal life, in multicellular animal life. We don't think they're important in plants. In plants, you have cellulose, and that's forming a similar sort of mechanical role in plants. It's making the cell walls and like the stems of plants and stuff, sort of like our connective tissues, which are made from collagen. Those cellulose is sugar-based, not protein-based. There are some evidence that bacteria and algae can make protein-like collagen-like sequences, but whether it's actually triple helical or more like gelatin, and whether it has any function, we don't know. Or at least I don't know, and I don't think others do yet.
I feel like you mostly answered this next question with that and this question's about why animals and not plants have collagen, and if there's some sort of mechanical property within collagen that makes it incompatible for plants.
Yeah, I'm not sure that it's incompatible so much as it just different things arose in evolution that fulfilled the need to sort of scaffold these two different types of multicellular life together. So the cellulose in plants and the collagen in animals. Um, yeah, I don't know more than that.
No, that's totally not your area of expertise, but it's an interesting question. Um, Already, oh, these questions are great. Thank you, everyone, for all these great questions. So somebody asks, say, after breaking my ankle, I was prescribed collagen supplement with magnesium, and while taking it, I noticed that an old injury that left surface veins dark and faded. Is is collagen the cause of that?
I don't know. I don't know. I'm sorry. There are so many reports of benefits of this, but a lot of them haven't been in super well controlled studies. So it's hard to know, and we're all different, and our bodies respond differently to things. So,
Okay, and this is maybe a similar answer, but just asking about is there collagen in our stomach linings, and can glycerin support in repairing the stomach lining?
yeah, I think there is collagen in the stomach lining. I'm If I had to guess, it's probably more of like the collagen for the network type of protein. There's another protein that's found called elastin that's on the that's also outside of cells. And as you can perhaps guess by its name, elastin, it provides a lot of extensibility to things. And so there are other proteins that are also contributing different mechanical features. Um, Whether glycerin has any role in helping cells to make their extracellular structures and assemble them, I don't know.
Okay, okay, we have a couple questions about the centrifuge and the spinning.
Uh-huh.
So what does the stretching of the collagen in the centrifuge have to do with our body's collagen and tendons? And then the other question, so you can kind of answer them together, is why doesn't the trypsin get shorn away from the field due to the spinning?
Yes. Okay. So let me take the second question first. So, I mean, I guess I have this little picture here, the schematic of what was going on with the cutting. So, trypsin will be affected by the forces too, but the issue is that it's so small and the density of trypsin is really not that different from water. And so the force that's exerted on it is not sufficient to sort of pellet it to the bottom. It's a few nanometers in diameter, and these beads are a few micrometers in diameter. So they're 1,000 times bigger. Plus, they've got little chunks of iron oxide in them to make them heavy and really pull down. So we are not affecting really anything in our sample chamber with this force, except for these big heavy beads. So we aren't sort of stratifying other things. As to the connection with tendon, thank you for asking. I guess I think about it again. I'm going to get my rope back here. And you probably can't see the little threads in this thing, but when I pull on a tendon, it's like I'm pulling on this rope. Okay. So I'm stretching it. How much of that force is being felt by each one of these little threads in here? I don't know, right? Clearly, they must be supporting some of this force because I'm not pulling this apart. But each little thread on its own is pretty weak. I could probably stretch it and pull it out. And so when we're doing the experiments on the individual collagens, it's like we're just taking the individual threads and we're measuring their response. And that's a really important part of the puzzle for understanding how the rope responds. But it's not everything because this just the little building blocks. And then we have to understand what are the forces that hold them together. And some of them may have some of these chemical staples so that when you pull them, they can't slide, but some of them might be able to slide past each other and sort of shear and come apart that way. And so there's more levels to this structure that we really need to be able to understand. One thing I can say is that people have pulled on tendons and we're starting to do this too, or at least the level down from a tendon, a fascicle. We've been doing some experiments in our group recently, and others have published this where you can stretch those. And what other people have shown is that as you stretch the fascicle, it does get cut more easily by trypsin, just like these single individual thread-like collagens do. Um, and it also seems to have some evidence that when you pull this whole structure, the individual collagens are unwinding. So we think there are connections, but to sort of say how much force is really on a single collagen protein inside of a tendon when you walk, there's a lot of assumptions that would have to go into that we're not able to really get to yet.
Awesome, thank you. Okay, we have a couple questions here about saunas. Um, I'll ask them both and then you can kind of answer them together. So, first one being, does regular sauna sessions help rezip your collagen? And then The other is if I get a fever or go to the sauna, does my collagen get affected?
Yeah, so those are really interesting questions. So I guess the first thing I want to make sure that I get across is that when we found the collagen falling apart at 37 degrees, that was the individual proteins. What I didn't say explicitly, perhaps, was that when they are in these higher order structures, so in a fibril that would make up a tendon, for example, if you heat your sample to 37 degrees, they don't fall apart. So if you want to break up one of these rope-like fibrils, you have to heat to a higher temperature, like 65 degrees. So the interactions between them really help to keep them stable. Now, on the surface of the ropes, this is what fascinates me, is they're still somewhat exposed to water. They're not all surrounded by their neighbors, making them really cozy. So I think there's probably responsiveness on the surface. So in a fever setting or a sauna or something like that, you will be in a hotter temperature environment. It's probably not too bad for your collagens that are in your tissues, at least unless you're in that vein for extended periods. I think there's a lot other things that are going on that your immune system's trying to fight disease or whatever if you've got a fever. Um, yeah, but I don't know, and I'm not sure that many people do. It's interesting because the when the collagen is first made, I talked about like, okay, we have to make each of these three strands. And then while it's folding or zipping up, you're also making some chemical modifications. It turns out you get more of those modifications at a higher temperature than you do at a lower temperature. So depending if you're growing cells in the lab and getting them to make collagen, you can control the melting temperature somewhat of the collagen by the temperature you grew your cells at. So, yeah.
That's super interesting, but I think we're probably all safe for saunas, so that's good to know.
Yeah.
Alrighty, so we have a couple more minutes, and I think in that time, if we have enough time, I would love to hear a little bit more about why vitamin C is important.
Okay, cool. Thank you. So this comes back to the same point I was just talking about, actually, which is these chemical modifications that get put on the individual chains, sort of while they're folding up. So one of the modifications is putting on an oxygen group or an OH, a hydroxyl OH group, on some of these amino acids, prolines, for those who might know that, to make them hydroxyprolines. If you don't put these OH modifications, these chemical modifications on, the chains will not fold. You will not get a stable triple helix. So it's absolutely essential to put these chemical modifications on in order to be able to make our collagen very stable, fold, get secreted from cells, form our tissues. So there are enzymes or these little machines that stick those groups on. And something that these need to do their job is vitamin C. So if you have vitamin C around, they can go make these chemical modifications. Your collagen will then fold up, get secreted from your cells, and make these connective tissues. If you don't have vitamin C in your diet, what happens is your cells can't make collagen properly. Okay, so think historically. Can you think of any time when there were issues with people not having enough vitamin C in their diets? Well, that was sailors back in the day, right? What happened to sailors? They got scurvy. Why did they get scurvy? Because they didn't have vitamin C. What happened to them? One of the first things that happened was their gums started getting all gooey and their teeth would fall out, right? So that tells you, ooh, my gums, they must need to make fresh collagen a lot. I mean, we can say this now, they didn't have any clue, but right. So that's an area where there's a lot of collagen that needs to get made. The turnover time is quick compared to what I was saying about like cartilage, which is really slow. So if you can't make fresh collagen, your body will be turning it over, using it up, like chewing it up and using those amino acids for other things. There's a natural balance between making and breaking. It's happening in bones all the time and so on. So if you stop being able to make collagen, your tissues will slowly start to fall apart at different rates in different parts of your body. So you need vitamin C to make good collagen. Now, if you think about the sailors, you go, whoo, how did those sailors get their vitamin C? Well, they got limes, right? The limeys, whatever. So when I learned about this, I was like, okay, cool. This is really cool. It connects to collagen. But then I thought, but how on earth do the people in North America get by, especially Canada, right? We don't have citrus fruits indigenous to our culture and so on. So how is this okay for us? Turns out spruce tips are really rich in vitamin C. And the indigenous people shared this knowledge with like Jacques Cartier and the explorers when they came to help them get through the winters and get their vitamin C. So I just think this is so cool. Yeah.
That's awesome. I'm so glad we had time for that. Um, I think we have time for one more thing, which is my favorite question to ask all of our presenters, um, which is what was the most surprising thing you learned in your research? Like what was just like something that really tickled you or surprised you in your research that you didn't expect?
I mean, I think the most fascinating thing that I've been involved with so far. What I love is the people that I work with. Like that's just what I love the most, discovering together. But I think this work on the collagen and actually seeing it fall apart and being able to characterize the rate at which this happens, and then Alla's work on refolding it. And I mean, I did not go into the details of this, but she was able to really pinpoint exactly where along this triple helix are the things that are sort of the break points and the challenges for this. And then she went and dug into evolutionary history and figured out, yeah, these staples are essential. It's been such a fascinating story, and it's so rewarding to see her bring it to life.
Um, I think just double checking questions. I think all the questions were asked. A couple of them were kind of put together because there were similar questions. Um, oh, yeah, I think we got through all of them.
Excellent.
Um,
They've been great questions. Thank you so much,
Incredible
everybody.
questions.
It's
So good.
lovely.
Um, but we are coming to the end of our time. So if you did miss the answer to your question, it will be available, recorded on the website. That should go up very soon. So you can always check back. Um, but I just want to say a big thank you to all of you for joining us this evening. I know I had an excellent time. I love learning new stuff about research. And if you do have any questions that come up, feel free to reach out via email, and I'd be happy to connect with Nancy on that. And thank you, Nancy, so much for your time this evening and for sharing your passion and your amazing research, both yourself and the whole group you work with. Um, we really appreciate it. And I hope that I see many of you back for our next Café Scientifique, which will be in April. We don't have one in March, but do look out for our Nobel Prize lectures, which will be in March. So thank you, everyone, and have a wonderful evening.
Thanks, everybody. This has been great.